So for all those who are so for all those who are new to my class, so Monday, Wednesday and Friday, so I teach a class 12 topic, and Tuesday, Thursday, Saturday, I teach for a class 11 topic. Alright, so today is Saturday and usually my class is from 4 to 5, but every Saturday 7 to 8 p.m. also I come live. I come with some interesting sessions or something extra. Okay, so previously we were talking about some motivational session, but now I'm coming up with a revision session. So because you guys suggested me to, so let's have a quick recap of your cell cycle and cell division, because always questions from this chapter are pretty confusing, right? How many chromosomes, how many DNA? We don't really understand. So once you understand the logic behind it, then it is not a really tough chapter. Alright, so shall we go forward? So before that, come on, quickly like, share and subscribe the session. So please let your friends know that we are having a session in YouTube. Come on, quick. So I give you 2 more minutes so that you can share my session, and then we will begin with today's class. So I'll try to finish off as quickly as possible because this is just a revision. Okay, so but I will try to revise the entire chapter. So in the next week, I'll bring questions from this chapter. Alright, yes. So shall we go forward? Come on, quick, do share the session. So please do like, do subscribe the channel as well. So Satyendra, hi. So I guess you are well before time. You have been waiting for my session. That's really great. That's really nice of you. Yes, Sweta, a very good evening. So after a long time, I am seeing you. So these days you seem to be very busy. So how are you doing? Fine? Good? But thank you so much for joining my session. So please do like, share and subscribe. So shall we go forward? Yes. A quick revision of cell cycle and cell division. So how many of you do you really feel this chapter is tough? Ma'am, even genetics, okay. So genetics is pretty tough. Even human physiology I can manage. But cell cycle and cell division, no, it's really tough. If you say you are very confident in genetics, then this chapter should not be tough because there is no logical correlation. If you are strong in genetics, then obviously you will be strong in this chapter also, because this also deals with your DNA. This also deals with your chromosomes, right? Yes. So don't give me illogical reasoning as to why this chapter is tough. So tell me where exactly you find difficulty in this chapter. So where exactly you want me to teach you again in detail. So there you can stop me. So now I'm going to teach you the entire chapter, a quick revision. I'm going to take you through all the topics, but in between, if you want me to explain any chapter in particular, any topic in particular, you can stop me right there. All right. So shall we go forward? Okay. So here we go. So weightage of this chapter. So this chapter comes under the unit structure and function. This is a class 11 topic. So the cell, the unit of life. So there are 3 chapters under this unit. So cell, the unit of life, then you have biomolecules, then you have cell cycle and cell division. I know I have not yet taught biomolecules to you, so I will be teaching biomolecules very soon. Alright, so since we have 3 chapters in this unit, and this unit contributes to almost 8% of your entire class 11, and cell cycle, so cell cycle only contributes to 2%, but still that 2% is tough for you if it is a numerical, right? Even if it's theory, you people will manage, but if it's a numerical, so that is where your base is not strong. But let me give you a quick recap. So cell cycle. So what are the topics that we're going to talk? So we're going to talk about cell cycle basically. What are the cell cycle regulators and the 2 types of division, mitosis and meiosis. Alright. So what is cell cycle? For example, all living organisms, so be it any living organism, so life starts from a single cell. So what is this single cell? This single cell is your zygote. Alright, so always your zygote is diploid. So remember this. Okay, so if you have 2 diploid organisms, they produce haploid gametes, which will in turn give you a diploid zygote. So a zygote is always diploid and a gamete is always haploid. So this is universal for all kind of living organisms. So don't ever get confused. All right. Yes, Anthony Abhi, hello, hi. So thank you so much for joining my session. Fine. So what is cell cycle? So when a particular cell divides, the DNA inside it replicates and the cell grows along this replication. So that is called a cell cycle. Fine. So the sequence of events by which a cell duplicates its genome, synthesizes the other constituents of the cell, and eventually divides into two daughter cells is called a cell cycle. So in layman terms, when I say a mother cell is giving rise to two daughter cells, so which means your cell is dividing. So these two daughter cells are identical, which means whatever is present inside your mother cell is getting shared between the two daughter cells, okay? So it has to duplicate its genome, even the genome has to be passed on, right? It also synthesizes other constituents of the cell. If this has mitochondria, both of these also should have mitochondria. So this process is called a cell cycle. So it is a sequence of events which include cell division, DNA replication, as well as cell growth. Fine. So this is how we represent it. So what are the different types of cell division? So when I said here in the previous slide, in the cell cycle, your cell undergoes division as well as your DNA replicates and your cell grows. So what are the types of division? So how does it divide? So we have three types of cell division: amitosis, mitosis, and meiosis. So amitosis, the process of mitosis does not happen. So simple mitosis gives you two identical daughter cells, that is mitosis. Okay, so here you do not have the formation of two identical daughter cells. Okay, so in meiosis, so we know mitosis is your equational division. So whatever is present in your mother cell will be exactly in your daughter cells. So your mitotic reaction produces exact copies of the parent. Alright, so in case of meiosis, if the mother is 2n, so when the mother cell undergoes meiotic division, it will produce cells which are n and n. So the genetic material is getting divided, is getting reduced. So meiosis is known as reductional division. So what is this if I write 2n, which means two set of chromosome? N stands for the number of chromosome. So when I say 2n, it is diploid set of chromosome. For example, we humans have 23 pairs of chromosome, right? So in case of humans, n is 23. So which means together we have 46 chromosomes, right? So that is what I am trying to tell here. Yes, meiosis is reductional division, mitosis is equational division. Alright, so now we will talk about each of them in detail. First is amitosis. So amitosis, so for the process of mitosis formation, you will have what is called a spindle fiber formation, division of chromosome, etc. But in case of amitosis, so you have no such formation. So it is the simplest mode of cell division. Which one? Your amitosis is the simplest mode of cell division. So what happens is that your nucleus elongates, it becomes dumbbell shaped, and it divides into two daughter nucleus. So the nuclear envelope remains intact. So if you have read this chapter before, so when your cell is undergoing division, first the nuclear membrane disintegrates. It disappears so that the nucleus, so the genetic material divides, right? So here the nuclear envelope remains intact. Alright. So such a type of simplest mode of cell division occurs in case of your PPLO, that is pluro-pneumonia-like organism. So blue-green algae, bacteria, and eukaryotic cell. So all these belong to your bacteria. So you can say they belong to kingdom Munidans. Okay, and this is your eukaryotic cell. Yes, sir, a very happy good evening. So thank you so much for joining us today. So please do like, share, and subscribe. Okay, so yeast, where it undergoes budding, it is a type of amitosis. So what happens? So you have your plasma membrane, you have your nucleus. So the nucleus gets elongated, it becomes dumbbell shaped, and the nuclear envelope remains intact. So this is your nuclear envelope. So this is remaining intact and it divides into two daughter cells. Alright? Yes? Okay, so phases of your cell cycle in case of your eukaryotic cell. So the eukaryotic cells divide once in every 24 hours approximately. So yeast is a unicellular eukaryote. Yeast is a unicellular eukaryote. It progresses through the cell cycle, the entire cycle, in 90 minutes itself. Okay. So two basic phases of a cell cycle are present. One is your interphase and the other one is your M phase. So M phase stands for mitotic phase. So interphase basically means the resting phase. Okay. So M phase starts, then it proceeds with the nuclear division, that is karyokinesis. So mitotic is basically the division of the nucleus. So division of nucleus technically is known as karyokinesis. So karyo is nucleus, kinesis is formation. Okay. Sorry, kinase is division. So karyo is nucleus, kinesis is division. So division of the nucleus. So division of the nucleus ends in cytokinesis. This is division of your cytoplasm. Fine. Okay. So interphase is the resting phase. The cell prepares itself for the division of your mitotic phase. So you have two phases in your cell cycle. The first is your interphase and the next one is your mitotic phase, that is your M phase. So for your M phase, the cell has to prepare itself. So that happens in the interphase. Though interphase is a resting phase, so now you can say your exam, NEET exam is on 1st of August, right? So now you're all in the interphase stage, but you're all active, right? So you're all in a quite resting phase because you do not have exam now. But it does not mean that you will stop preparing, right? So similarly, all your cells are in the interphase stage where it is resting. It is preparing itself for the big day, that is the mitotic phase. Alright. Hi Priya. Yes, good evening. So thank you so much for joining. So in interphase, you have three subphases. So G1, S and G2. So these three phases put together, it is called as interphase. Alright. Fine. So your G1 phase, it is called as GAP1 phase or growth phase. Okay. So S is the synthesis phase where your DNA is getting synthesized. G2 is the growth phase, is growth after your DNA replication. Then M is your mitotic phase. All right. So before we actually go into the cell cycle, so this is a cyclic stage, right? It starts with your G1. It goes to your S, it goes to your G2 and finally your M phase. So this is the direction or order in which it proceeds. What if the cell, when it's preparing itself for the mitotic division, is not completely prepared? So for example, some of you might be half prepared, but you still will appear for the exam to give a try, right? So, but in case of cell cycle, no such chance, factor or probability is taken into account. Okay, only if the cell is completely satisfying all the criteria of cell division, only then it undergoes division here. Okay, otherwise it has to be stopped. There has to be someone to guide you, right? So if you're studying or not, you have your parents at home to guide you, to regulate you. So similarly, if a cell is fully ready for synthesis or not, so you have cell cycle regulators which tells us. Okay, so regulation of cell cycle. So there are factors whether or not to move forward through the cell. So there are factors which decides whether a particular cell will move forward or not. Okay, yes. First, I will revise the concept and then in the next class, I will revise only questions because both I will not be able to finish because to finish the revision of entire chapter, I will need 1 hour. So that is why I have not got any questions here. But while teaching, pay attention to the concept. I will teach with reference to your questions. Okay. So, so these factors, okay, they include both external clues as well as internal clues. Okay. So external clues like your molecular signals which come, which say stop the cell division process. You also have internal cues like when you have DNA damage. Okay. So if at all a mutation happens, so what happens then? You, for example, if in a particular cell in the DNA there is a mutation, then there will be regulators which will say take the cell out of the cell cycle process. Okay. So cues like these, they act by changing the activity of the core cell cycle regulators inside the cell. Fine. So everywhere you have a checkpoint which regulates your cell cycle. Fine. So how many checkpoints we have and who are the checkpoints? So one type of cell cycle regulators is your cyclins. Okay. So cyclins are among the most important cell cycle regulators. So cyclins are a group of related proteins. There are 4 basic types found in eukaryotes and in other humans. So what are the 4 types of cyclins? You have G1 cyclins, G1S cyclins, then you have S cyclin and M cyclin. So please remember we have 4 types of cyclins, G1 cyclin, G1S, S cyclin and then M cyclin. Alright. Okay, so each cyclin is active only during a particular phase, or it is active only for a transient period of your cell cycle. So not all the 4 types of cyclins are active throughout the cell cycle, only at a particular duration they are active. Okay, for example, when you talk about M-cyclin promotes the events of your mitotic phase. Fine, such as your nuclear envelope breakdown and chromosome condensation. Fine. So you have these phases, so only at certain stages the cyclins are active. Fine. Then apart from cyclins, you also have one more class of regulators. They are called as cyclin dependent kinases. So we commonly call them as CDKs. So cyclins and CDKs are the most important type of your cell cycle regulators. Fine. So cyclins drive the event of cell cycle by partnering with the family of enzymes that is known as cyclin dependent kinases. So cyclins alone cannot regulate your cell cycle. So along with them you have other players as well. The other player is your cyclin dependent kinases. So when either cyclin can perform alone or CDK can perform alone. So both have to be present together to activate the process of regulation. Alright, so CDKs are basically kinases. Whenever it ends in ASE, remember it is a class of enzyme. Fine, so these are group of enzymes that phosphorylate your specific target proteins. So phosphorylate means when they attach a phosphate group. Fine. So see, here you have only your CDK. Okay, there is no cyclin. So without cyclin, your CDK is inactive. Okay, so there is no turning on of your cell cycle regulator. But if your CDK and cyclin are present together, it helps in phosphorylation of your target protein, and thus your cell cycle proceeds. Only if the regulators are active, your cell cycle will proceed. If at all your cell cycle regulators are not efficient enough, then you have uncontrolled division of your cell, and that is when that happens in case of your cancer cells. In case of cancer cells, your cell cycle regulators are absent. Alright, so remember that much. Okay, so here we go and talk about each of the different phases of your cell cycle. So first is your quiescent stage or your G0 stage. So some cells in your adult animals, they do not exhibit to divide. Example, your heart cells. They divide only once. They do not repeatedly divide like your skin cells. They have divided once. Now they do not want to divide. If at all there is an injury, if at all there is a need, only then they divide. Okay. So cells of these kind, okay, they divide very rarely. So they exit your G1 phase because this is the order. G1, yes, G2 and M phase. If they are not in need, if they do not have to divide, they exit your G1 stage and enter your G0 stage. So all cells which are capable of division will enter your cell cycle phase. Okay, but in the G1 phase, it will be decided whether there is a need for the cell to undergo division or not. If there is no need for this particular cell to undergo division, it will enter into a quiescent stage, that is your G0 stage. Once when there is the need, so from your G0 stage, it will go back to your G1 phase and continue with your cell cycle. Alright, fine. Okay. Now, phases of the cell cycle. So, we are going to talk about the interphase. So, interphase is the phase between two cell division. So, what happens? So, you have interphase is followed by meiosis. Again, meiosis is followed by interphase, right? So, it is a phase between two cell division process. So, you have three phases, G1, S and G2 phase. So, it is also known as the resting phase or the preparatory phase. Fine. So, it is the longest phase of the cell cycle. So, it takes 95% duration of your cell cycle. So, this means the cell, so in your preparatory phase, your cells are metabolically active because it is taking care of its needs. For the mechanism of cell division, what are the substances that it needs? It is trying to prepare itself. So, the cell is metabolically active right now. So, if you look at a cell in the interphase stage, you can see the nucleus, the nuclear envelope very intact, okay, which means the cell is metabolically active here. So, talking about your G1 phase, so G1 phase is the most variable phase. Why? Because once a cell enters your G1 phase, then it decides whether it has to undergo division or it has to undergo quiescent stage. So that is why it is the most variable phase. So here your cell growth occurs. So your mitochondria, chloroplast, lysozyme, ribozyme, endoplasmic reticulum, Golgi vacuoles are produced. So if a mother cell is going to divide and give rise to two identical daughter cells, then there has to be two mitochondria, right? So your mitochondria, your chloroplast, your lysosomes, ribosomes, endoplasmic reticulum, Golgi vacuoles are to be produced, right? So structural and functional proteins are also produced here. So the metabolic rate of your cell is pretty high in this stage. So this is also known as pre-DNA synthesis phase, because this is just before your S phase, right? So in your G1 phase, so this is how your cell is. Your cell is preparing itself for the cell division or the next phase. So the next phase in your cell cycle is your S phase. Your S phase is the longest phase in the human interphase. Okay. So the longest phase of your cell cycle is your interphase. So in your interphase, you have three substages, G1, S and G2, alright. So among that, your S phase is the longest phase. Yes, ma'am, a very happy good evening. So thank you so much for joining us. So please do like, share and subscribe. Fine. So the replication of DNA takes place in this phase. So protein molecules called as histones are getting synthesized here. So once again, now you guys will tell me what is the DNA, what is a gene, and what is a chromosome. So if you get these terms clear, then this chapter becomes clear. Now come on, quick, tell me what is a DNA, what is a gene, and what is a chromosome. And also you will tell me what is this and what is this. So what is the difference between these two structures, and what is the distinction or the difference between these three terms? Yes? So which comes before, which comes first, or which is present inside what? So do you have any clue? Yes? So I guess everyone knows what is DNA. So DNA is your genetic material that contains the information that is being passed on from one generation to the next, right? So our DNA is double stranded like this. Fine. So our entire DNA does not produce protein. So what is the role of DNA? To carry the genetic information. If the genetic information from your mother is getting passed on to you, it has to be expressed, right? For that, it undergoes what is called a central dogma of life. So your DNA is copied on into your mRNA, and your mRNA produces your proteins, right? Yes? Fine. Now, any sequence on your DNA that codes for your proteins is called as a gene. So you have your entire DNA does not code for a protein. So only the sequence on your DNA that is coding for a protein is called as a gene. Okay, so your DNA is too long, it cannot be directly put inside your cell. So the length of your DNA is 2 meters and the diameter of your cell is in micrometer conditions. Thus, it has to be compacted. Thus, your DNA is packed inside chromosomes. So that is called as a chromosome. So now tell me, so inside your chromosomes, you have your DNA, and any sequence on your DNA that codes for a protein is your gene. So now you got these three terms clear? Yes? Fine. Now tell me, what is the difference between these two? So I know this is a chromosome. Then what is this? And I also know, this is a chromatid. Fine. But what is the difference between the structure 1 and this structure? Between this and this, what is the difference here? Yes, any idea? What is the difference here? Understand, so these are the chromatids of a chromosome or the arms of a chromosome. So this one is called as your centromere. So this is a chromosome. This is also a chromosome. Alright. So though this has only 2 chromatids or 2 arms and this has 4 chromatids or 4 arms, both are called as chromosomes. Hi Shimpi, hello. So thank you so much for joining us today. So please do like, share and subscribe. Fine. So this is a chromosome. This is also a chromosome. Then what is the difference? Wait. So whenever the chromatids are attached to the same centromere, it is still called as a single chromosome. So this is also one chromosome, this is also one chromosome, because the chromatids are attached to the same centromere here. So in your G1 phase, so pay attention, in your G1 phase, your nucleus will have chromosomes like this. So in your S phase, where you have replication of the DNA will be doubles. So here if you have two chromosomes, first chromosome and second chromosome, even in S phase you will have only two set of chromosomes but twice the amount of DNA. Here how many DNA you have? So suppose initially imagine you have 2x DNA in G1 phase. In S phase you will have 4x DNA, but the number of chromosomes will remain the same because this is how it doubles. So if this is chromosome 1, this is also chromosome 1, only it is getting replicated. So the two chromatids are attached to the same centromere. So that is why they say the chromosome number remains the same, only the amount of DNA increases. So you have questions, right? If the initial amount of DNA is 2x or 2c, then what will be the amount of DNA at the end of G2 phase? It will be twice, because in S phase the amount of DNA doubles but the chromosome number remains the same. Is this clear? So please highlight this point. So in your S phase the amount of DNA doubles but your chromosome number remains the same. Okay? Yes, exactly. So here you have two chromosomes, here also you have two chromosomes, just that the DNA has doubled in your S phase. Is this clear? Yes. So you will not make a mistake in questions of this type. In cell cycle and cell division, so one type of question which they commonly confuse students is between the amount of DNA and the chromosome number. So once again the chromosome number remains the same. If the mother has 23 pairs of chromosomes, here at the end of S phase also you will have 23 pairs of chromosomes but twice the amount of DNA. Alright, is this clear? Fine. So DNA is packed with the help, so your DNA is packed into histone structures, right? So before we reach your chromosome structure, your DNA is packed in your histone molecules. So packaging of DNA, so your DNA is a lengthy molecule, right? So it has to be compacted inside your cell. So the length of your DNA is 2 meters. So what happens? And your DNA is negatively charged. So thus you have positively charged basic proteins known as histones. So histones are rich in arginine amino acid and lysine amino acid. So both of these are your basic amino acid. So what happens? So histone is a group of proteins. So histones are made up of arginine and lysine. So you have 4 types of histone: H1, H2A, H2B, so you have 5 types, H3 and H4. Fine? Okay. So H2A, 2B, H3 and H4. H2A, H2B, H3 and H4. So these 4 types of histones, yes, into 2 numbers each. So when I say 4 into 2, it is 8. So they form a set of octamer. So that is called as histone octamer. Okay? Yes. So this histone octamer is like this. And your DNA is wound around your histone octamer. So that is why you need your histone proteins. Fine. Now you understood what is a histone? Yes. So when your DNA is wrapped around your histone, so where does this H1 come is this. So between 2 histones and the DNA wrapped around, so this is where your H1 histone comes into picture. So H1 histone is a linker histone. Alright, is this clear? Fine. So a group of histone octamer which is wrapping your DNA is known as your nucleosome. This structure is known as nucleosome, and repeating structure of nucleosome is called as your chromatin. And this is further arranged into your chromosome structure like this. So that is why in your S phase, it is synthesizing your histone proteins as well. Alright. So protein molecules called as histones are synthesized here. Fine. So the centrosome replicates in the late S phase. So what is the centrosome? So long I have been talking about a centromere. So centromere is the one that holds two chromatids together. So this is a centromere. So centrosome, we'll talk about it in the process of mitosis. Alright. So this is your S phase. Now talking about your G2 phase. So we are done with your G1 phase. Then we have done with your S phase. Now we are talking about the G2 phase. Fine. So the tubulin proteins are synthesized here, which helps in your spindle formation. So this is also known as post-DNA synthesis. Then the cell stores ATP here, because for the process of cell cycle and cell division, for the process of mitosis, ATP is needed. So thus the cell stores or reserves ATP for itself. Okay. Hi Keshav. Acha, you were playing Menti. Alright, that's okay. So hi Shilpa, hello. So thank you so much for joining my Menti. So Keshav and Shilpa, so please do like, share and subscribe the session. Fine. So this is your G2 phase. Now finally entering into your M phase. So the number of chromosomes in the parents and the progeny cell is the same, because M phase here talks about your mitotic phase. Okay, fine. So this is your equational division. So the mother cell gives rise to two identical daughter cells. Okay, hello, hi. So you didn't come for 2-3 lectures in between. Why? What happened? Very busy? That's okay, but thank you so much for joining now. So please do like, share and subscribe. Okay, so karyokinesis involves 4 stages. So especially in your mitosis, your nucleus is going to divide. So karyokinesis means division of your nucleus. So here you have 4 stages. So you will remember it as PMAT. You'll remember it as PMAT. Okay, so prophase, metaphase, anaphase, telophase. So it happens in this order only: prophase, metaphase, anaphase and telophase. Alright, so now we'll talk about each of this phase in detail. So prophase is the first stage in your mitotic division. So what happens is your chromosome material condenses. Okay, so initially inside your nucleus you will have material like this. So at the initiation or at the start of your prophase, you will be able to see structures like this, called as, which are visible as chromosome only in the prophase. Okay, yes. So the completion of prophase. So how do you know the prophase is completed? Is when the chromosomal material condenses to form a chromosome. Okay, so you will have 2 chromatids attached together at the centromere. So centrosome undergoes duplication. So what is this centrosome? So centrosome is needed. Okay, so centrosome was produced in your late G1 phase, right? Sorry, in the late S phase. So your centrosome is something that pulls your chromosomes apart. Okay, so the centrosome that has undergone duplication during an interphase, now it begins to move to your opposite poles. So hold on, if you don't understand centrosome, I will explain. Okay. Then, so we'll talk about what are asters. So 2 asters together with the spindle fibers form mitotic apparatus. So hold on. So during prophase, so what happens? Your nucleus is going to divide. So that is your mitotic phase, right? So the first phase of your mitosis is your prophase. So in your prophase, what happens? The nucleolar membrane first disintegrates. Now because your nucleus is only going to divide. So once it divides, all the other things can be formed again. So hold on if you're not getting. So M phase is your mitotic phase. So in your M phase, okay, your nucleus is going to divide. So this is known as karyokinesis, division of your nucleus. So what is present inside your nucleus is your chromatin material, right? So we are going to divide this chromatin material, because so long in the S phase only the DNA has doubled. Okay, yes. So now we are going to divide this chromatin material so that it gives rise to 2 daughter cells. Fine. So if the chromatin material has to divide, first the nuclear envelope that is surrounding your chromatin will disappear. Okay. So now only this will be present. then what will happen? This will become visible as chromosomes in your prophase. So this is what happens in your prophase. The nuclear envelope will disappear and the chromatin material will be visible as chromosomes. Fine? Yes, Shimpi, did you understand this much? Okay, hold on. Now, so during the prophase, the nucleolus also starts disappearing. So if you remember the structure of nucleus, so what it contains? It contains nuclear envelope, and the center portion, it also contains nucleolus and your chromatin material. So even your nucleolus will disappear, only your chromatin material will be present in your prophase. Fine? So your centriole separates and moves towards the opposite pole of the cell. So you have something like this which are called as centrioles. So centrioles basically try to pull the chromosome apart from each other. So if you have a chromosome like this, now this has to be separated into two daughter cells, right? So now this has to be separated like this. So they will be pulled in each corner or in each poles. So who will pull the chromosome is your centriole. Okay, so centriole is a cylindrical structure. There are two cylindrical structures, if you can see like this. So these centrioles are produced or duplicated in your interphase, but only during the prophase they move to the respective poles. Can you see, this is one pole, this is the other pole of the cell. So around the centrioles you have astral fiber like formation, right? So this is called as astral rays. So when your centriole, this is your centrosome. So this is your centromere. Okay, so this is your centrosome. So centrosome contains your centrioles. Fine. So these are your centrioles. Fine. So surrounding the centrioles, if you have astral-like spindle fibers, it is known as astral rays. Fine. You're getting what are centrosomes and what are astral rays? Yes. Okay. So now, based on the type of meiosis, the astral rays can be present or not present. So listen, an astral, which means astral rays are not present around your centrosomes, around your centrioles you can say. So in case of plants, there are no asters formed. So in case of animals, asters are present and they are called as amphiastrals. So if you look at here, so you have your centrioles. So this two centriole structure together is called as a centrosome. Alright. So surrounding that you have an astral-like fiber. So this has astral rays. But here you look, so there are no centrioles at all. So plants do not have centrosome or neither centrioles. So they are anastral. If you ask me then, ma'am, is it possible to undergo cell division? Yes, it is possible to undergo. So in plants, it is adapted to undergo cell division without your centrioles. Okay, so just like some people have hair conditioners at home, so during summer they turn on, but it's not that the other people who do not have air conditioners die, right? So they adapted to living that way. It is somewhat similar to that. Okay. Okay, so next after your prophase, it is your metaphase. So complete disintegration of your nuclear envelope. So everything in your nucleus disappears except chromosome. That is your metaphase. Okay, so the morphology of chromosome is best observed during your metaphase. Okay, so the chromosome is made up of two chromatids. They are held together by a centromere. So the one that is present in between your chromosome is your centromere. So this is your centromere. Fine. So you have small dish-shaped structures in your centromere like this. So these dish-shaped structures are called as kinetochores. So kinetochores is where your spindle fibers attach. So your spindle fibers connect the centrosome to your centromere. So on the centromere, it is attached to your kinetochore centers. Fine. So the plane of alignment of chromosomes at metaphase is known as metaphase plate. So all the chromosomes lie together at the same plane. So the alignment is done. So this is called as metaphase plate. So can you say you have your centrosome, the centrioles are here, astral fibers are formed. So these are attached to the kinetochores of your chromosome. Yes. So this is how it happens. So when a particular cell is dividing, so can you see red chromosome and blue chromosome? So one is from the mother, one is from the father. So that is why you have 2 chromosomes. If I say we have 46 chromosomes or 23 pairs and we have 23 chromosomes, right? So we have 2 copies of each chromosome, one from father, one from mother. That is why we have 46 chromosomes. So that is all the chromosomes are getting attached here. Okay? Yes? Fine? So the next phase is your anaphase. So Shimpi, are you getting? Did you understand? So the next one is your anaphase. The centromere splits and the chromatids separate here. So the chromatids move to the opposite poles and your anaphase is the smallest phase of your cell cycle. So in the previous diagram, you saw they were still stuck together. But in the anaphase, they are getting separated. They are being pulled to the opposite poles so that now this will become one set of chromosomes. This will become the next set of chromosomes. Alright? Yes? Yes, centriole plus aster. So basically centrosome is only your centriole, but if astrals are present, it is called as astral rays. Okay? So this is what happens in case of your anaphase. Now the last one is your telophase. So in your telophase, the chromosomes cluster at the opposite spindle poles and their identity is lost as discrete elements. So initially I told you in prophase, the chromatin material is present like this. Then in your prophase, it becomes visible as chromosomes. So in your telophase, once again after separation, it is like this, right? In your telophase, it is like this. Now what happens in your telophase? Once again, it becomes like this. It loses its identity as a chromosome. Fine. So now once again, a nuclear envelope forms around the chromatin material. So now you have 2 daughter nuclei formed. So now the things have been separated here and here. Yes, so which means 2 nucleus is formed. Only the cytoplasm has to divide. The nucleus has already divided. So can you see the nuclear envelope is getting rearranged? So all the other organelles are getting reformed. Nucleolus, Golgi complex and ER are reformed. Alright, fine. So now the next stage after karyokinesis, it is your cytokinesis. Fine. So what happens here is separation of your cytoplasm. Only when your cytoplasm divides, the 2 cells are have reached the complete division, right? Yes, so the animals is the formation of cytoplasm is different. In plant cells, the formation of cytoplasm is different. So how it happens? In animals, you have the appearance of furrow in the plasma membrane. So in case of animal cells, so remember animal cells have no cell wall. Right? So it has only the cell membrane. So the cell membrane is rigid. At 2 points, it forms a restriction. It bends at 2 points. Okay. So this forms a furrow. So when this forms a furrow, it forms a contractile ring. Okay. It forms the pre-line for division and then it divides into 2 separate daughter nuclei. Okay. In case of plant, it is your cell plate formation. Yes, you are right. So plants have cell wall. The plant cell wall is rigid, so that is the function of the cell wall. So the cell wall is providing rigidity to the plant cell. Yes. So now since your plant cell is rigid, it cannot undergo bending like this, right? So what happens is at a particular plane, you have Golgi vesicles being attached. At a particular equator or a particular angle or in a particular plate, your Golgi complex get attached at one plate. At one plane. So this arrangement of Golgi vesicles during the cell plate formation of your cytokinesis is known as phragmoplast. Fine. So this phragmoplast then will separate and it gives rise to 2 daughter cells. Now our cell has completely divided into 2 daughter cells. Is this much clear? Yes. Are we done? Fine. Now, we have what is called as modification of mitosis. So normally when, yes, calcium pectate forms the middle lamellar layer. Okay. So normally mother cell divides to form 2 daughter cells. But there can be some modification here. So cryptomitosis of promitosis. They are the primitive type of mitosis. Initially, this is how mitosis happened. That is cryptomitosis. Here what happens, the nuclear membrane does not disappear. You have an intranuclear spindle. Intranuclear is within the nucleus you have a spindle, because the nuclear membrane is not disappearing. So within the nuclear membrane you have a spindle. So that is why it is called as intranuclear spindle. So the division of amoeba by binary fission is a type of cryptomeiosis. So they are the primitive type of mitosis. The next type is dinomitosis. It happens in your dinoflagellates. So what happens here? So dinoflagellates are your mesokaryotes, because they are the connecting links between prokaryotes and eukaryotes. So that is why they are known as mesokaryotes. Alright. So here the histones are absent. So chromosomes fail to condense properly here. So the nuclear membrane keeps persisting, but the chromosome fail to condense, but a spindle is formed. Okay. So this is your dinomitosis. The third one is your free nuclear division. So only your nucleus divides, but your cytoplasm does not divide. So that is called as free nuclear division. So this is, you have division of your nucleus, but your cytoplasm is not dividing, which means inside one cell you have many nucleus. That is your coenocytic condition. So example is your endosperm, because endosperm has 3 nucleus, right? Fine. So the next one is endomitosis. So duplication of the chromosome without the division of the nucleus. Now the entire nucleus does not divide, but only the chromosome keeps dividing. So which means if I have 46 chromosomes, inside the same nucleus your chromosome keeps on dividing, but the nucleus does not divide. So this leads to polyploidy, increase in the number of chromosomes. That is called as polyploidy. Then endo-reduplication. So it is a modification of your endomitosis. Okay? Yes? Fine. So now what is the significance of your mitosis? So mitosis is restricted to your diploid cells only. Okay? So only when it is 2n. So the daughter cells will also be 2n. But even your haploid cells can undergo mitosis. Mitosis is equational division. If a haploid mother cell undergoes division, it's going to give you haploid cells. So in case of higher plants, your diploid cells undergo mitosis. In case of lower plants and some insects, only haploid cells undergo mitosis. So here the mother and the daughter cells are genetically identical to each other. Alright, so this also restores the nucleocytoplasmic ratio. What is this nucleocytoplasmic ratio? So in a normal cell, if I have the cell like this and the nucleus should be like this, right? So there is a particular ratio or the size between the size of the cytoplasm and the size of a nucleus. Suppose if my nucleus is like this, this is abnormal. So under a microscope, when you observe these two cells, you can say this nucleus is abnormal. So you can say this is a cancerous cell. So to restore the nucleocytoplasmic ratio, your mitosis has to be proper, which means your cell cycle regulators has to work properly. It has to be coordinated. Alright, so these are the significance of your mitosis. Then of course mitosis happens when there is cell repair. When a particular cells or group of cells are not present, to replenish the cells, mitosis happens. Alright, yes. So I know I will not be able to finish so much. So I have only taught mitosis because you people had a bit of doubt. So I think at least till mitosis everything is clear. So I will come back another evening where I will teach about meiosis and I will solve questions together. Alright, is that fine? So for now I will stop here. Hello, what happened? You're coming so late. Huh? You were there before the class started and you're coming when I'm going to say bye. Alright, I'm not starting meiosis because meiosis will take more time where I have to explain again things properly. So another class maybe on Tuesday evening again 7 to 8 we will meet. So where I will teach you about meiosis and we'll also solve numerical based questions from your cell cycle and cell division. Is that clear? Yes? Fine. So thank you so much. Okay. Okay. All right. Fine. Okay, Satya. That's okay. Fine. So thank you so much for being a wonderful audience and joining me on a Saturday evening. So hope you guys have a very great weekend. Okay. And one more thing. So Monday, please do join me at 4 o'clock. So we are going to start reproduction unit in your class 11. The last unit in your class, in your class 12. All right. Yes. Thank you so much and stay tuned to get more notifications about my schedule. All right. So bye-bye and take care.